If you're struggling or starting A-level biology, I'm an examiner for 3 different exam boards. I've helped hundreds of students personally get into medicine, 30,000 hours of tuition and teaching in A-level biology. I've helped hundreds and thousands of students get A's and A stars and grade 9s from GCSE to A-level. So stick around and we're going to start the first topic, which is basic components of living systems. So this section is 2.1, and in this section we're going to cover 3 main parts. We're going to cover the use of microscopy, on everything from how to use a microscope, and then how to prepare slides and how to observe slides. Next video we'll go through a common PAG technique, which is going through biological drawings. If you're just starting A-level biology, well done, you've picked the best topic. And you'll see that there's a big jump from GCSE to A-level, but there's a lot of common topic areas that you can build on. To start the topic off, we're going to begin with cell theory. Now the good news is you don't have to memorize all the names of these scientists, but I've summarized lots of them for you. What you need to first of all understand is the way that cell theory is an example of scientific theory. And what we mean by that is when we get a new observation and new technology, over time we come up with new theories and then we prove them and we experiment on them. So the very first scientist to ever observe a microscope was Robert Hooke. And he looked down a microscope and observed cork, and he called those little round things cells. And since then, lots of other scientists have built on that information, and as technology, as scientific equipment and microscopes have advanced, we now understand what's inside of cells to a really good level. This is an example of demonstrating scientific theory, which goes as new evidence is gained, we disprove or correct or adjust old theories and new theories form. So it normally requires an advancement in technology or advancement of understanding or a new observation, and from that observation we disprove old science and go to new ones. Now all of these resources are completely free. If you go to www.stemcelltuition.com/free workbook, you can download all of my module 2 workbooks completely for free with built-in exam questions, all of the slides and revision cards summarized with thousands of multiple choice questions. Click the link and you can download it for free. So the first thing we're going to learn is how we actually prepare a slide and how we use a microscope. So let's start on the microscope. So the first thing that we have to understand is how a microscope works and why is the setup the way it is. So there's 2 main regions within the microscope that we're going to first observe. You have 2 lenses. You have the eyepiece lens and you have the objective lens. And if you've used these in school, you're probably aware that you can adjust the objective lens and the eyepiece lens is normally stable, although you can remove it and put a new one in. Now the reason why this is set up this way is to reduce this phenomenon called chromatic aberration. Light is made up of many different wavelengths. Some of them are longer, some of them are shorter, and as a result of that, when we observe light we see a mixture. When light goes through a lens, that causes chromatic aberration, and that essentially means the light goes blurry and it spreads out, and it's the failure of the lens to focus all the wavelengths at the same point. And by setting it up with 2 lenses, we reduce the chromatic aberration. Now we also need to know some of the key points of a microscope that you remember from GCSE. So we have the coarse and fine adjustment knob. They both do the same things. They just move the stage up and down. Because when you look down the microscope, you're actually looking at a specific speck, a specific place, and you're raising the slide up or lowering it down, so your focus is exactly on the cells that you want to observe. We have the stage, and we'll be looking at things like stage micrometers later on. A really important point to know is our objective lens. I use the formula TOW as a way to work out the total magnification. So the total magnification is the magnification of the individual objective lens multiplied by the eyepiece lens. So for instance, if the objective lens is on 40, the eyepiece is on 10, we times them together to get a total of 400. Most of the time for A-level biology, you'll just be given magnification as the total, but you need to be able to calculate it just in case. So you can quickly now have a go and see if you can label all the parts of the microscope. Good work, let's move on. So now we know the basics of a microscope. We need to know how to actually prepare something to observe, because most of the time if you just put something on the microscope it won't be blurry and you won't be able to see it. So these are the 4 ways for A-level biology in which we prepare a sample. Now in school you'll mostly do wet mount, squash, and maybe some smear slides in terms of blood, but you need to be aware of all 4 types. So the first type is simply dry mount. That's where you take a slide, you put whatever you want on the slide, so this is meant to be a hair, and then you observe it underneath. Sometimes you might do a cover slip, and I would recommend you always mention in your exam answers to say that you're going to put a cover slip on, even if it's not in terms of moving air bubbles and water out of the way. The next example is a wet mount. Now in a wet mount, the reason why you're doing it is because you want the cell in its normal natural environment. So what we do is we normally use a pipette and we put some water within the cell. We then place our sample cells in the wet area, and then we place a cover slip on the top. Now the cover slip you normally place on with something called a mounted needle, which is just a long thin needle with a plastic handle. And you do that at 45 degrees and you slowly lower it down because you want the air bubbles to escape. One of the most common misconceptions students have is they see circular things that they're viewing and they think it's an air bubble. Now a squash slide is almost identical to this, but the difference is once you've got it prepared, you're going to physically push down on the cover slip. And you're generally going to do that for a couple of reasons. The first reason is normally to move things around within the cell. It might be that the chromosomes you're trying to observe are pushed down at the bottom side of the cell, so you might squash it down and that will push the chromosomes close to the surface for you to observe. And the final version, which generally takes the most technique, is known as a smear slide. And the PAG, the blood smear PAG, you may or may not do in school, and you can even view your own blood. It's really interesting. You can take a small pinprick, you place your blood on the slide, and then you smear it and you can observe your own blood, and you'll see the red blood cells and the white blood cells within it. Now the technique for this is slightly different. You want an extremely thin smear, because red blood cells are extremely thin themselves. An average red blood cell in terms of its width will only be about 7.5 micrometers. So therefore the height of it is even shorter, and you want to try and get it evenly spread and only one cell thick. So we use a smear technique to do that. So to do that, you do a small drop of blood on one side of the slide. You then use a second slide, and then you drag across the blood, and as soon as the blood comes into contact with the slide, you'll see it will smear along the inside of the second slide. And then you're going to drag that slide across, and as you drag that slide across, somewhere around about here you'll end up with a really thin section. Then you can focus on that section to stain, stick to the slide, and then you'll hopefully get a thinner and more easily viewable slide. So now we've done the basics of sample preparation, we're going to go into more specifics of why are we doing it, and we need to learn about bacteria. First of all, why are we staining in microscopy? Well, you've probably seen on screen they have low contrast, so we want to increase that contrast. If you look at the cells on the image here, that they're generally quite blurry, and there's two types of microscopy that you'll do in A-level biology. The first is the normal way you've done at GCSE, which is known as bright field microscopy, where the light comes up through the specimen and you want it as thin as possible so as much light can get through. Or the alternate version is the light is coming from above. Generally it's a lower magnification with a dissector microscope, and you just look down on the specimen. Normally you do this with things like insects such as fruit flies that you'll do in year 13, or you might do it to view larger structures like striations in muscle of chicken wings. Either way, both have a low contrast. So what we do to overcome that is we stain them. Now there's one particular type of staining that we're going to learn in the most detail, and that's known as differential staining. Now staining allows us to see a contrast, the difference between two places. What we're trying to stain is individual cells. Can we tell the difference between them? So actual types of cells. And then within a cell, so this is a root cell doing some mitosis, I want to see the individual organelles, such as the chromosomes here lining up for mitosis. So we stain for two reasons: between types of cells and within cell organelles within the same cell. Both of them increase the contrast. Now there's actually three marking points here: types of cells, cell organelles, and contrast. So write all three when the question is about differential staining. But if the question, for instance, is about cell organelles, so inside of a cell, you'll probably only need to mention that one. Now we also need to know the process of staining bacteria, and there's two types of bacteria: gram-positive and gram-negative bacteria. Gram-positive, once stained, which you have to learn the process of. So the first stage of staining bacteria is to add a stain called crystal violet. Then you're going to add another stain, iodine solution, and it is a solution because it's potassium iodide, not pure iodine, which is a solid. Then you're going to use alcohol. You'll normally do something like ethanol, which is the most common one you'll use, and that's to fix the cells to the slide so they stay there. Now, gram-positive bacteria will appear blue-purple, and you can write both in the exam. The way that I remember blue as a very positive colour, like the seaside, like calm, like sky, so blue goes positive, whereas gram-negative bacteria should stain red. Now, often you will stain some bacteria and you might not see all of them there. So you have to do the same process for gram-negative, but then you add a counter stain known as saffronin. That should appear red, and that will be negative. Now, the way that I remember negative is I think of negative as negative things, as in fire, as in anger. Now, there's a reason why they stain differently, and it's because of the way that the cell walls are built up. So you don't need to learn for A-level biology the individual structures of the cell walls, but you do need to know that they're different, and as a result of them being different, they react differently to different antibiotics. So gram-positive bacteria exposes its main cell wall component, which is peptidoglycan. Those on different exam boards will often see the phrase murin instead here. So you need to learn that peptidoglycan is the cell wall type of bacteria, which is different to plants, which is cellulose, and fungus, which is chitin. But gram-negative bacteria have this like extra outer membrane that can interfere with antibiotics. They also still have a peptidoglycan, but as you can see, the structure is different. We don't need to learn the structure of the cell membranes of bacteria. Now, on top of that, which is easily confused, so positive and negative bacteria is easily confused with positive and negative staining, which is something different. Positive staining is where a stain particle attracts to something within a cell. So an example of this might you have a negatively charged cytoplasm attaching to a positively charged stain particle. Now you'll see in this image here of some lung tissue, it's the actual lung tissue itself. These are the cells of the lungs. So this is the inner lining of a bronchiole. And the actual cells themselves are stained. And most of the time you can assume you're doing positive staining. But we also do a separate type of staining known as negative staining. And that's where we actually stain around the outside of whatever we are observing, and that creates a difference in contrast. So the best example of this you can see here is these are negatively stained bacteria called cocci, which is spherical bacteria, and the stain is being repelled because the cell itself will match the charge of the stain. So some examples of this are Congo red, and this makes it stand out against the background. You'll soon be ready for your first A-level biology exam question, and I'm going to talk you through some general exam technique that will serve you throughout your years in A-level biology. So first of all, when you look at a question, you want to spend the majority of your time breaking it down. So if you've got a 3-mark question, I'd recommend a whole 3 minutes on figuring out what to say and 1 or 2 minutes to write it down. In A-level, you get longer in the exam than you actually have marks. So let's break it down. So the question is describe. So when we see a describe question, that normally means we want the what, the when, and the where in steps. So what is being used, when is it being used, and where is it being used? So we're trying to describe the steps of how to use the equipment. So when it says that in a question, you want to explicitly mention all of the things they have given you in the question. You'll notice, for instance, here there are 2 slides, so you'll need to mention that. So how do we prepare a blood smear that can be stained and viewed? And your next technique is, most biology A-level questions come in 2 parts. There's a before and an after, a stained and a viewed, often like describe and explain. So expect that in your answer. And your final technique for today is, if you see 3 marks, it's so easy at A-level to miss answers. And at A-level, they'll often be part of a mark scheme, then a comma, and then a second part of the mark scheme. At GCC, that might have been enough for 2 marks, but A-level, it's not. So you should always aim for more marks than there are in the question. So you have a go now, pause the video and see how many you get. Okay, well done. Hopefully you've got full marks here and a quick breakdown of the mark scheme. So you had to physically mention the word. When it's underlined, you have to use that exact word. So for instance, here it says use the pipette to place blood on the slide. You needed all of that for 1 mark. And by explicitly mentioning that we place it on one side of the slide got you the 2nd mark. Then using the slide or the cover slip to drag it across, to spread the blood across the slide. So if you said, for instance, use the slide to spread, no mark, to spread the blood, and I would avoid it in this situation. If you say it here, now blood is not underlined, but there's a good chance the examiner won't give you the mark. Just mentioning that you're going to do it at an angle to reduce air bubbles is also valid. And any other general staining technique will also be valid here. Good work, that's your first lesson done. In the next video, we're going to apply this in terms of how to do a biological drawing, which is really common in your early PAGs that most students fail. So click the link, go into the next video, which is how to do biological drawings. See you there.